basically the epidemiology, right? Kind of who, what, where, when, why, how, all that fun stuff. And then some of the basic pathology, basic treatments, current and future. All right, now keep in mind, because of the power of Zoom, if there's someone that's on Zoom that you would like to work with, right? Zoom people don't just have to work with Zoom people. Lecture in-person people don't have to work with just lecture in-person people, right? You can kind of do a mix or a combo of those things, okay? Hey, Ajax, can you text my wife and see what she wants? She's trying to call me. I just want to make sure it's not an emergency. So just be thinking about that. And remember, I think I mentioned this way back when, but I reserve the right to nix a disease. And typically if I nix a disease, it's not because I don't like you or I don't think the disease is interesting. But there are some diseases that have had, I don't know, 50 cases throughout the history of the world that we know of. And that means there's just not a lot of good information out there on those diseases, right? Again, I'll put this in email, but if you're trying to decide what to find, like if you World Health Organization neglected tropical diseases, those are always some good ones, right? One thing I've learned from teaching this class is that you don't want to get a disease while you're in the rainforest or jungle. They seem very unpleasant, all right? But there's some other sources. So again, think about who you're going to want to work with. I'll send out a reminder email today and think about what disease you want to work with, right? It'd be good if you're on the same page as your partner, right? I don't want Abby working with Alanis and Alanis is like, I want to do the plague. And Abby's like, I want to do hantavirus. Like, okay, well, then as friends, you need to work on your communication skills. All right, so just keep that in mind, right? I'll send out a reminder email for that. Exams, I just finished grading an A&P exam, so now my focus turns to you. So my dream of dreams would be by Wednesday evening, all right? But Alyssa, what happens to dreams? Yeah, that's so hard, but so true, right? But I will try to have those done by Wednesday evening is going to be my goal. That way, if I miss, I'll at least be close to that and hopefully have them done in under a week for you guys. So just be patient with me there. All right. Any prayer requests? Yes, ma'am. What's his name? Jesse Buckner. All right. So we'll pray for Jesse for flight school. Cool. All right. Anybody else?
I think I feel like a lot of people are just kind of tired this semester.
Preach it, sister. I never realized how much I appreciated that 2-day fall break until right now. I'm just like, professors are tired, students are tired. All right, professors that are tired and students that are tired, when they come together, results in some type of explosion that might be worse than the nuclear bomb, right? Okay, good. So I'll just pray for maybe energy and kind of a second wind for the semester, right? This is your only full month of school, or last full month of school though, right? All right. Okay. Because I don't know, to me it's kind of motivation to help me make it. All right, 1 full month, that's it. All right, 1 full month left. All right, so energy and resolve for the semester. Jesse for flight school. Anybody else? All right, let's pray and then we'll get started. Lord, just thank you for this day, this opportunity to be here in person. We thank you for your provision throughout the semester. We thank you that for the most part, the precautions that Lee has taken have allowed us to make it to this point, and I just ask that you help us finish the semester well. I specifically just ask for energy. I ask for a second wind for students and faculty and staff that are kind of feeling the grind of the semester without that typical break. And I just ask that you help us finish well. I ask that you help us show each other grace as our energy levels are running low. I also specifically today lift up Jesse Buckner as he gets ready for flight school. I ask that you show him favor, ask for protection for him, and ask that this experience would be what he has been hoping for and what he's been wanting. And we look forward to when he is finished and is certified. And Lord, I also just continue to lift up all those that may be dealing with COVID-19 or other illnesses and ask for healing for their bodies. For those of us, for those that are well, I ask for protection. And I just also continue to lift up any unspoken prayer requests that may be in these students' hearts. I thank you for loving them and hearing them. In your son's name we pray. Amen. All right. I know the anticipation has probably been killing you, but it just is going to make it that much more enjoyable. Rachel, would you like to do it from your seat? Sure. Okay. So Rachel's going to present her current event article, and then we're going to jump on in.
Okay, so the title of mine was Customizable Synthetic Antibiotic Outmaneuvers Resistant Bacteria. So basically, they were looking at antibiotic resistance, and they wanted to see if they could, instead of since making a new antibiotic.
Yeah,
It could take decades, and that's a lot of money. They wanted to see if they could revive some of the older antibiotics that bacteria had already been resistant to, which sounds a little weird, but trust me. Okay. So they wanted to break down the antibiotics and sort of use like molecular Legos to find out like a core combination that they could use to then like add on to or take away so they could make larger molecules. So they demonstrated this approach using a class of antibiotics called streptogramins. These kind of disable the bacteria by, they said, like kind of gumming it up by attacking the bacterial ribosomes so they can't make proteins. So the bacteria that are resistant to this they're called Virginiamycin acetyltransferases. They just call them VATs in the article though. And so they kind of grab the drug before it gets in there so it can't attack the ribosomes. So they wanted to see a way to sort of evade that whole process to make the antibiotic, like the VATs can't even tell that the antibiotic's there so that it can just go in and kill it. They chose streptogramins because they used to be really effective in fighting Staph aureus, which is, that infection is kind of common. So yeah, they started out this process and they wanted to make 7 molecular models that they could kind of tweak as they went along. And those were like the core models. And so they had different like groups of 7. And so they used cryo-electron microscopy and X-ray crystallography to make 3D pictures of the drug, the ribosomes, and the VATs so that they could see the interaction that was happening there. And so they wanted to find out which parts of the antibiotic were essential to the function. And so they looked at how it fit together and how everything, the process that it went through. And so they found like the core one that they thought, okay, yeah, this is what makes the antibiotic work. And so they kept that essential group together, and then they started switching out different parts to see if they could modify it to where it would still work, but the VATs couldn't like recognize it. So they tested it on some resistant strains of Staph aureus, and they found out that it was 10 times more effective than other streptogramins. So that's really cool. I mean, it's not like huge yet because it's still, streptogramins are still, bacteria is still resistant to it right now, but they found that through all this studying, and like the 3D models and stuff, they could see, okay, well, the bacteria could possibly use this mechanism to become resistant. And so they found that they could go like 2 steps ahead and go ahead and make the antibiotic to where the bacteria won't have the opportunity to become resistant through that potential mechanism. So I thought that was really cool how they can now like sort of redesign old antibiotics that we thought were just kind of useless at this point. And I think it's really important as Christians because we're supposed to be stewards and we're supposed to take care of people. And this is potentially like a way to start curing more infections because we now have the ability to sort of revive and remake antibiotics. Any questions?
Okay, you might not know this, but you said they are doing this because it's really expensive to make antibiotics for like.
Yeah.
But do you know if the molecular Legos are expensive?
Yeah, they didn't talk about that. I don't know, because, I mean, yeah, like all the tests and stuff. I'm not sure, but.
Did they say, like, how long? So maybe they tried it?
No, they just said it was groundbreaking and it was the start of being able to really like these synthetic antibiotics that they could really make them not resistant to anymore.
Okay. All right. Well then, let's jump on. Ashley, lights, please. I mean, I've only been once, but huh? See that? Yeah. I mean, I think it's gonna be helpful. You know, I can move so much better, but I still have, you know, there's still like discomfort and pain. So I'm hoping by the time I'm 40 to be back to normal. So I got five years, right? 5 years. All right. So today, and well, I'm not making this material up. The order I'm kind of adjusting a little bit because I'm doing the class in a different order than I've ever done before. And so I kind of thought this seemed appropriate because we've talked about pathogens, we've talked about the immune system and antibiotics some. And so I kind of wanted to start to touch on, well, what are some of these differences between prokaryotic cells and eukaryotic cells? And again, we've mentioned these at least when we talk about effective antibiotics, right? Targeting the pathogen versus the host. And another thing, you know, that we'll go over is microbial growth. And that'll be I think the next unit that we do. And that is just really relevant to lab, right? And you'll see the implications that has with what you're doing in lab. And again, it also has an implication in medication and your immune system because of the rate that these, I was gonna say guys, but guys or girls, right? If we can assign sex, I guess, equally to things that don't have it, right? But they can go quickly and rapidly, and that has effect on epidemiology and public health. That has an effect on how you treat them. Has an effect how your immune system interacts with them. And so this is just kind of maybe putting a little bit of a bow on this. And I'm hoping too by the end of the week, Professor Perry, who you guys all know from lab, is the fungi expert, all right, or the fungi whisperer. I like that one better. She's the fungi whisperer. And so I'm going to see if she can come in either Friday. If she can't come in Friday, maybe even next Monday, to talk about fungi a little bit. One of the things that gets neglected in this class some are microbial eukaryotes, right, which fungi would be one of. And so I think Professor Perry does a great job of doing it justice. All right. Now, you know, some of this is going to be review for you guys for a couple reasons. One, because you've seen it before. in a previous biology class. And two, even if you haven't had much prokaryotic basic biology, one thing that you will see is that there are a lot of similarities, again, and you guys know this, between prokaryotic cells and eukaryotic cells. Now, what we're going to primarily focus on, or what I'm primarily going to refer to, in this case, when I talk about prokaryotic cells, are bacteria. Archaea also have a lot of similar structures. Remember, through genetic analysis, we actually have put these in different domains, right? So you have eukarya, prokarya, and archaea. But again, structurally and functionally, archaea and bacteria are very, very similar. So I'm going to kind of treat them as one. All right. So let's start to talk about prokaryotic cell structure. All right. All prokaryotic cells, right, have a cell envelope, right? So all the bacteria that you're working with in lab have a cell envelope. And you guys know this now, hopefully, right, that they all have a cytoplasmic membrane, very similar to you. All prokaryotic cells have a cell wall. Again, all is a dangerous word in biology because we'll talk about a couple exceptions, right? But for the most part, we'll say they all have a cell wall. And then where you start to see a really big variety here is in a capsule, all right? And so a capsule, right, is going to be this outer extra layer that's kind of again, a sugary gel-like material. Okay. So let's talk about what you should know and what you should hopefully remember from previous biology classes is that the cytoplasmic membrane of a prokaryotic cell is made of phospholipids, right? Just like your cell membrane, right? Remember, it has a hydrophilic and a hydrophobic region, right? So a region that hates water and a region that loves water. And basically, when you look at this phospholipid bilayer, right, you're able to keep things on the outside out and things on the inside in, right? So it's a really nice structural design. And so, again, we see here, or we will see here, and we're not going to talk about it some, but there are a lot of similarities. And this is one of the reasons, if we're talking about antibiotic drugs, that there's not going to be a lot that are targeting the cell membrane, right? Because they're going to have that phospholipid bilayer, just like our membranes. And just like our membranes, right, they have things that can get across via simple diffusion, right? They also have transmembrane proteins, just like we do. What's a transmembrane protein? Can someone remind me? That's had a biology class before. Right, it spans through the bilayer, right? So it's basically a touch point or a connection point for the inside of the cell to the outside of the cell for things that can't easily cross the membrane, right? Now you'll see here in just a second, really interestingly, in some ways, right, I would say that prokaryotic cells actually rely more heavily upon these transmembrane proteins than even you or I do, right? Again, we're not going to go big on it, but those of you that have had I guess, is it cell bio, right, Michael? Is that where you guys talking about this stuff? Cell bio, then you've probably heard of the fluid mosaic model. This shouldn't be overly shocking to you, right, that a cell membrane or a cytoplasmic membrane is not just some hard shell, right? Proteins can move within that bilayer, right? It's kind of a free-flowing structure, all right? In some cases, right, we allow certain gases to cross, water and other small hydrophobic molecules. Remember, it's just advantageous if things like oxygen, nitrogen, and CO2 can cross the membrane without needing a transport. So again, you're seeing the same basic concept in prokaryotic cells and eukaryotic cells, hopefully emphasizing to you the idea that a cell membrane is not, right, a common target for antibiotics. Okay. What we also see here is that prokaryotic cells, like eukaryotic cells, are subject to osmotic forces, right? Solute concentrations. So some of you guys did we do, I'm trying to remember if you guys would have done, did we do any of this in bio? Yeah, we did. Did you guys do the eggs in bio 1? Did any of you take bio 1 way back when with Riley? Oh, no, I guess we changed it up before you guys did. We used to do this cool experiment where if you take eggs, you soak them in vinegar, it removes the brittleness of the outside shell. And then you're left with like it almost feels like a water balloon. And you can put them in hypertonic and hypotonic solutions, and then you can weigh the eggs to measure movement of water. All right. So anyway, what's really kind of cool here, though, is that you see, right, if you have a hypotonic solution, right, that means the solute concentration is higher on the inside. Again, I know some of you guys are pros, maybe some of you guys hate this type of stuff. So please remember, one of the easiest things to think about as solute is think about a sugar concentration or a salt concentration to maybe put this in perspective. All right. Remember, we like balance. And so water flows in depending on the permeability of the membrane, right? The solute can also flow out, right? So it can be a two-way street. In this case, though, right, you have a swelling here, and the cell wall actually provides a little bit of extra, I guess, resistance or structure to that. A hypertonic solution, okay? You can see here the solute concentration is high on the outside, so we pump a bunch of water out. When we do that, all right, the cytoplasmic membrane, right, and the cytoplasm shrinks, right, because you're getting rid of all that water. Now, you know, one of the reasons that this is important, and not that you guys do this a lot in lab, but what's one thing that you don't put your bacteria in if you want it to live for a long time? A common liquid that you shouldn't put your bacteria in that's readily available in your lab. Water, right? So if you put bacteria in water, right, water, hopefully, and I haven't tested it from our taps, should not have a high solute concentration. So water should be hypotonic, right? And so that bacteria is going to take in a lot of water, and then it's going to burst, all right? Is studying bacteria that burst a good idea? No, I don't think it is, right? But to each their own, right? And so these basic osmotic principles, again, that apply to your cells are also going to apply to prokaryotic cells. Again, the interesting thing is, in some cases, that cell wall can provide them a little extra protection, right, for their structure. All right. Again, we're not going to spend time here because you guys should hopefully remember this, right? These are some of the transmembrane proteins that Michael talked about. Transmembrane proteins are pretty convenient because two of the things that we use them for are facilitated diffusion and active transport. Please remember, if we talk about diffusion, high concentration to low concentration, right, those molecules flow freely. This is simple diffusion. It can go through that phospholipid bilayer. Facilitated diffusion does not require energy, all right, but it does require a channel to allow those molecules to go across. And then active transport, right, requires energy because you're going against a concentration gradient. Again, you guys have seen this before. Now, where this starts to get interesting, all right, is because of a cell wall, right, the concept of acquiring nutrients via something like endocytosis, right, is not a great option in bacteria, all right? So what we see in prokaryotic cells is that digestion of their nutrient source takes place outside of the cytoplasmic membrane, all right? Now, it's true, you guys can also digest things, right, on the outside of your cytoplasmic membrane and then bring them in, right? We secrete enzymes. If you're talking about the digestive system, right, you secrete a lot of enzymes that then break down nutrients that are outside of the cell so they can take them in. But you also have the capability of taking in nutrients via endocytosis, right? So bigger kind of macromolecules, right, you guys can easily consume or your cells can easily consume. That's not the case, right, for prokaryotic cells. So prokaryotic cells, what they have to do, right, is they make a protein. And what's really cool, right, in this case, the protein or an enzyme, right? Remember, all enzymes are proteins, but not all proteins are enzymes. Right? Food for thought. So what's really interesting, though, too, is that you don't want this enzyme assembled inside of the cell, because if I told you you're not capable of endocytosis, well, then you're also not really capable of exocytosis. And so you have to assemble the enzyme that's going to digest the nutrients on the outside of the cytoplasmic membrane. Now, right, this is probably not a new concept to some of you that have had cell bio in the past, and maybe you even talked about this in genetics. I can't remember, but what we see on some protein molecules that help them, or protein peptides, I'm sorry, that help them get to where they need to go, right, is they have a little signal sequence. So in this case, these enzymes have a signal sequence that designates them, right, to pass through a transmembrane protein and be secreted into, well, you'll see, not really extracellular space, but outside of the cytoplasmic membrane. That enzyme can then assemble. It can break down, right, the macromolecule that this bacteria wants as a nutritional source, whatever that may be. And we'll talk about some different options there in a little bit, right? It's broken down into these small subunits. These small subunits can then easily be transported, right, across another transmembrane protein. All right. So you have external digestion, right, outside of the cytoplasmic membrane before, right, those nutrients are brought into the cell. And, you know, depending on how much you love evolutionary biology, right, I have a paper for you if you're interested that actually talks about a species of bacteria that's kind of a little bit of an in-betweener. It looks like it has some modified abilities to do endocytosis, right? So it's kind of like that gap species, if you will, right? But again, prokaryotic cells in general are not doing endocytosis or exocytosis, and so they rely heavily on external digestion of their nutrient source. What's also really interesting, and you'll see this, and I can't remember if Professor Perry will touch on it in her lecture. If she doesn't, I might supplement a little bit. This is also the same way that fungi are going to digest, because fungi also have what was a structural feature that fungi share with prokaryotic cells? Does anyone know? Cell wall, right? So again, that cell wall, right, makes you know, bringing in things via endocytosis, exocytosis very challenging. So fungi are also going to secrete enzymes that digest their nutrient source. All right. One of my favorite things, I'll be honest, when I was like your guys' age 2 years ago, no, is anyone buying that? I'll shave and try to look young like you guys. But when I was a little bit younger, right, I really hated the electron transport chain. I just hated it. And now, man, I just love it. All right. And what's really interesting about the electron transport chain, again, are the strong similarities between prokaryotic cells and eukaryotic cells, but then these really kind of, in my opinion, kind of wacky, weird, fun differences. All right. So if you remember, right, the basic concept of the electron transport chain is you are pumping, right, a bunch of protons out, right? So you're basically making your own concentration gradient. You have a bunch of protons out here. There's no protons in here. Protons rush in, right? Those of you that remember, they rush in through something called ATP synthase, which basically looks kind of like a windmill. It spins around and it pops phosphates onto ADP. All right, you can tell by that description. That's one of many reasons that I'm not asked to teach chemistry courses here, right? But that's the basic gist of it. All right, and so Holmes, can you remind me, where does this happen in your cells? Mitochondria. Yes, we love the mitochondria, right? So you have your cytoplasmic membrane, right? It has its transmembrane proteins in it, its phospholipid bilayer, maybe a little cholesterol in there for structural rigidity, that those of you that remember that, all right? But your cytoplasmic membrane doesn't have anything to do with the electron transport, right? But we have double membrane bound organelles. So your mitochondria, right, has its own essentially phospholipid bilayer in it, right? And that's where electron transport takes place, right? So we call, the mitochondria the energy house or energy factory, whatever you want to call it, of the cell, all right? Or you can even call it, the battery of the cell, right? Also, those of you that go into grad school, professional school, nothing makes people more frustrated that do mitochondrial research when you call it the battery of the cell. So you can file that one away if you want to kind of get under their skin a little bit. What's really cool, right, is that this process, right, electron transport, right, the electron transport chain is actually taking place in the cytoplasmic membrane of the prokaryotic cell, right? So their cytoplasmic membrane has, right, these proton pumps and these other electron transport proteins embedded, right, throughout the whole cell, right? So if I'm telling you that a mitochondria is a battery, in this case, then you would say, well, the whole prokaryotic cell could be considered a battery, all right? One of the things that we'll kind of touch on here, right, they don't have these membrane-bound organelles like you guys have, right? So that whole individual cell, right, has to have the capabilities to do everything, right, that your little organelles specify, all right? What we will also talk about that's really interesting, and we'll kind of get to this, I think, in this lecture and later, is that prokaryotic cells use this proton gradient to actually power their flagella, right? So they can make their flagella spin, right? Which allows them to get to places. I think you guys, even in fact today, no, wait, not today. Even in fact tomorrow, I think Professor Perry made them this weekend, we'll be doing a motility auger, right? And bacteria are motile to have flagella, right? It's kind of like a spinning motor. You'll see that here in just a little bit. So we use the proton force for that. They can also use the proton gradient to help actively transport things, right? So to transport things against their proton gradient. So in you guys, the proton, right, gradient that you generate via the electron transport chain is used to make energy, right? ATP. In prokaryotic cells, first of all, right, it's in the cytoplasm, which is different than yours. It's used to make ATP, just like you. You don't have flagella, all right? What's the only cell in humans that has flagellum? Sperm, all right? And although functionally it serves the same purpose, right? Structurally, it's actually very different, all right? And then, right, active transport, so moving things against their concentration gradient. You're using ATP, they're actually using the proton gradient to help them with that, right? So you guys use the electron transport chain for one thing. These prokaryotic cells actually use it for 3 different things, right? So kind of a cool, unique feature, all right? So let's talk about cell walls, all right? I've mentioned these some with antibiotics. You guys have done gram-positive and gram-negative. How did your guys' gram stains go? Were they beautiful? Yeah. Professor Perry would speak highly of you. Caitlin, would she? So? Yeah. Okay. Gram staining, whew, it can be a little tricky, right? It can be a little finicky, right? But Professor Perry probably went over it. The basic principle, right, is that essentially gram positive, right, a thick peptidoglycan cell wall are going to retain that crystal violet dye a lot more easily than gram negative, right? One of the things that you treat these cells with when you do your gram stain is a solution that was high in alcohol, right? What does alcohol do to a cell? What were you disrupting? It's okay. I'm tired too. I'll just rest in the awkward silence to gain more energy. Did you guys do PEA plates yet? No? What is alcohol doing to a cell? Why is alcohol bad for a cell? What dissolves in alcohols? Lipids. What is the cell membrane made of? Lipids. Woo, right? So when you are washing bacteria with alcohol, you are disrupting, right, lipids. You'll see here in just a second, gram-negative, right, cells have more lipids in them.
When you do, what kind of stain is that when you wash it with alcohol?
Will you do that?
Don't you do that, or is it, I'm thinking of decolorizer, never mind.
No, decolorizer is an alcohol.
Okay,
base.
so then how does that not disorient the gram-positive and gram-negative?
Oh, Blair, that's a good question that I have a great answer for, right? So if we're actually talking about gram standards, we can talk about the basic principles. Remember, one of the things you did, does anyone remember what you treated your cells with after crystal violet? Iodine, right? That's why it didn't work, Rachel. So you treat it with iodine, right? So iodine actually causes cross-linking between molecules of crystal violet, so it makes it harder for them to leave the cell. So there's a couple different things going on there, right? So crystal violet goes inside the cell, right? Iodine helps it cross-link. When you treat with alcohol, you do a couple things, right? You start to punch holes in the cytoplasmic membrane, right? So crystal violet could leave. But you also, the way I kind of look at it is you almost turn the cell wall into a little bit of like shrink wrap. So you dehydrate that cell wall and it starts to kind of compress. And so that helps hold on to the stain. All right. Now, and I'll come right back here. This is, I just want to show it to Blair's point. So this is a gram-negative, right? Its cell wall is much thinner. And so your shrink wrapping, right, of that cell wall when you treat with ethanol, right? And so a good decolorizer is ethanol with a little bit of acetone. It's just not as effective at holding that dye in, and right, that alcohol is punching a hole in this outer membrane because it's lipid based along with the cytoplasmic membrane. So it's just easier to poke holes in gram-negative because they have more things that are easily disrupted by the ethanol. Does that kind of help a little bit? All right, so let's take a quick step back, right? Peptidoglycan, right, is made of two sugar subunits, right? NAG and NAM, right? N-acetylglucosamine and acetylmuramic acid. Please feel free to call them NAG and NAM, or you can memorize the whole name, right? Up to you. Those of you that have taken ANP1 had a neurology unit where you realize that the number of possibilities for synaptic connections in your brain is almost limitless, which means that your memories that you can form is technically almost limitless. And so if some of you want to test that, right, please feel free to memorize a little bit extra. But for me, NAG and NAM is just fine. What I want you to notice too is that these sheets of NAG and NAM alternate. I'm sorry, these subunits. So you go like NAG, NAM, NAG, NAM, NAG, NAM. All right. And then you'll see here in just a second that you have sheets, you stack these sheets on top of each other, right? And it's actually going to be NAM molecules on kind of parallel sheets that are connected to one another, right? So my hand is a NAM, right? My arm has a NAM on it, right? So that's kind of the connection point between those parallel sheets, all right? You're not connecting NAGs of parallel sheets. You're only connecting NAMs of parallel sheets. And you'll see here, I'll come in on this in just a second, all right? So again, this is a gram positive, a thick peptidoglycan layer, all right? You can see here, you have this gel-like kind of area or space. Again, so this is gonna be where digestion of nutrients takes place, right? So outside of cytoplasmic membrane, but actually technically, right, still inside of the bacteria, right? Because it's in between the cell wall and the cytoplasmic membrane. You also have these little red guys here, teichoic acid. Teichoic acid provides some structural stability, but as far as we know, its most important function is that we think it actually kind of acts as a, we'll say like an ion magnet, right? And an ion magnet in this case is important because a lot of enzymes that you use and a lot of enzymes that prokaryotic cells use require ionic cofactors. And so we think this is helping bring in some of the ions that make enzymatic function possible in prokaryotic cells. Now, this should not be surprising to you, right? That this thick peptidoglycan layer in a prokaryotic cell, in this case gram-positive, is permeable to sugar, amino acids, right? Basic nutrients. And you want those basic nutrients to be able to get to the cytoplasmic membrane where they can then be taken in, right? It's pointless to have a shell on the outside if things can't get by it for you to survive and live. So this is actually showing you what the connection looks like between those parallel sheets of peptidoglycan in a gram-positive cell. So here, right, what you see is the presence of something called a peptide interbridge, right? So you basically have indirect linkage of NAM molecules. So here you have a tetrapeptide chain, two tetrapeptide chains, this intrapeptide bridge. The reason that I point this out is because in a gram-negative cell, the tetrapeptide chain connects directly to the adjacent NAM molecule. All right, so the way they're kind of stuck together is actually slightly different between a gram-positive and a gram-negative. All right, so this is absent in gram-negative, this intrapeptide, or this peptide intrabridge, right? The tetrapeptide chain in this case would just go directly down to an adjacent NAM molecule. All right. Please note, I've seen, on exams, if you're talking about a sheet of peptidoglycan, right? Note, right? You have the NAG, NAM, boom, right? They just those alternating subunits there. But again, remember when you're talking about connection of parallel sheets that are stacked on top of each other, whether it's gram-negative or gram-positive, right? It's going to be NAM molecules that are linked together, right? You're not linking, right, or attaching NAM to NAG or NAG to NAG when you're talking about the parallel sheets. This one is positive. Yes. So in the negative, you would just have a tetrapeptide bridge that comes directly down. Right, so they have, I would say, a direct linkage of parallel sheets. For gram-negative, gram-positive have this indirect kind of linkage of their parallel sheets. All right, we already talked about these different components on the previous slide, but again, this is kind of for visual reference, all right? So please take this in, right? Let it sink in. This is gram-positive. If nothing else, you should be able to look at this next image, even if you know nothing about microbiology, and tell me, Professor Daft, those look very different. And I would say, unknown student, you are correct, all right? There are some key structural differences that I hope jump out to you right away. I already pointed some of these out to Blair. One of the first ones, all right, is that gram-negative bacteria have this crazy wild looking hair, right? And that crazy looking wild hair is actually something called lipopolysaccharide, right? Or LPS, okay? And we'll talk about LPS here in just a second, right? Another thing that you may note is that the peptidoglycan layer in gram-negative bacteria is much thinner, right? I pointed that out when we're talking about the gram staining. You also have, all right, these big yellow guys here called porins, right, which are transmembrane proteins that are through this outer membrane, right? You also have lipoproteins, right, that span this membrane and then even interact with the cell wall. Okay? And although similar in kind of function, it is slightly different, right? Remember in the gram-positive, if you take a look one slide back, you had that kind of gel-like layer that existed between the cytoplasmic membrane and the cell wall. Well, here you have something called a periplasmic space, which is kind of the equivalent, right? Except it is a kind of a gel-like layer that exists between the cytoplasmic membrane and the outer membrane of a gram-negative cell. And within it, right, you have the cell wall. So when we talk about something like, and we'll hit this in just a second again, right, something like penicillin, right, which loves to interact with cell walls. One of the things I hope you can appreciate, right, and we've mentioned this, but now maybe you can see it, is that penicillin is not going to do very much against a gram-negative bacteria, right? A couple of things. Penicillin interacts with cell walls. Well, this is a really small cell wall, a thin peptidoglycan layer. 2, right, in order for penicillin to even interact with this cell wall, it has to get through all of this stuff right here. All right, so this is kind of this outer membrane is an extra layer of defense, right, that can be kind of tricky for us to deal with when it comes to antibiotic medications. Okay. So let's talk about LPS really briefly. Some of you, right, depending on what you do and later in your lives may have heard this referred to as endotoxin, right? Remember we talked about pyrogens, right? Things that induce fevers in the inflammatory response way back when. Well, LPS is a great pyrogen, all right? So LPS is the reason that gram-negative bacteria might induce a fever in you. LPS has 2 major components. You could go back and look at this previous diagram. It has the O antigen, right, which is the outer portion. So that was that fun looking hair, right, which is variable amongst bacteria. So you could actually, right, on a molecular level, look at LPS and distinguish different gram-negative bacteria from one another, all right. And then lipid A, this is the portion of LPS that is anchored in that outer membrane. This is, right, that highly inflammatory component. So if you have a gram-negative infection, if your immune system is killing gram-negative cells, right, this lipid A is released. That's the pyrogen. It induces an inflammatory response. In a laboratory setting, we actually will take LPS, right? You can basically buy it in powder form. You can add it to a cell culture to induce inflammation and mimic activation of the immune system. So it's a really, really useful molecule, all right? So as I said, right, used in research, right, it's helped us discover important cytokines and chemokines, right, signals by cells in your body that activate the immune system. And so it's helped us, right, in rheumatoid arthritis and Crohn's disease, right, so autoimmune diseases. And you guys hopefully recognize this, right, pathogen-associated molecular pattern, right, a PAMP. So LPS is a PAMP, right, that interacts with TLR4, right, which is a pattern recognition receptor, which you guys hopefully all know and love. And look, it could appear again on an exam. So if you didn't get right the first time, redemption opportunity is yours. All right, so I'm going to end here, right? We'll pick up talking about porons, right, which are an important component of the outer membrane of gram-negative bacteria. We'll start that on Wednesday. I will be sending out an email again today about your disease presentations. Again, those won't happen till November, but I want to give you, right, a nice stretch of time to work on that stuff because like Blair said, right, we are feeling the burn right now. So I want you to have plenty of time and not have to rush that later in the semester. All right, get out of here. Have a great Monday. And I